From each of these partners today, and we'll hope to leave some time for questions.
Okay, I just need to make sure I know how to work this. And is this? Okay. Well, hello everyone. My name is Timira Gajanov. I represent the Ovid Therapeutics. Many of you probably heard about the company. It's a small biotech company. We are based in New York, and we have a couple of people working out of Boston. We've conducted the first Angelman study, which was completed last year. I'll talk about the study a little bit later. And we're getting ready to start the phase 3 study. So, how does it? This is the disclaimer. So just a little bit more about Ovid. So we have two molecules that we're working right now. One is the OV101. We're doing studies in Angelman as well as the Fragile X. And also we're conducting studies in rare epilepsies in collaboration with Takeda, which is our sponsor and partner. And we're doing studies in rare forms of epilepsy. So what we do, we completed the phase 2 study called STARS. It was completed last year. The goal of the study was really to establish the safety of the molecule as well as the efficacy of the molecule. And we are confident enough that we have a very favorable safety profile, and we have enough efficacy data to start the phase 3 study. We spoke to the FDA a few times, and we got a blessing to start the phase 3 study, which is about to start. At the same time, we have an ongoing study, ELARA. It's an open-label study. Every single participant who completed the STAR study is now able to enroll into the ELARA study. It's an open-label extension. So I think everyone who participated in the STAR study knows about the ELARA, but if you have a loved ones who were patients in a STAR study and do not know that ELARA is now up and running, please do share this news. We have the same sites for ELARA as we had for the STAR study, and the study is going to be ongoing for a couple of years. Neptune, phase 3 study, we are about to start. We are conducting the site feasibility, we're finalizing the protocols and completing all the regulatory documents. So hopefully later this year we're going to have first patient screened. So a little more details about the phase 3 study. It's a multi-center study. It's randomized. It's going to be double-blind study, placebo-controlled study. The study is going to be global. I'm going to describe the sites that they have been selected or are being selected for the study, and the goal of the study to evaluate the efficacy of the OV101. Study design. The patients are going to be screened, and they're going to have 4 weeks to complete the screening procedure. At day 1, if they satisfy the inclusion-exclusion criteria, they're going to be randomized into either placebo arm or the OV101 arm. The treatment period is going to be 12 weeks, and we're going to be conducting the different assessments as the study progresses. They're going to be 4 or 5 face-to-face visits with your PI, with your primary investigator. But most of the visits are going to be conducted via phone. We're going to have approximately 15 sites globally. We're going to have sites in US, Israel, Australia, Europe, and the site population is mainly 4 to 12, but we will have some patients that are younger than 4. So patients who are 2 to 4 years old, we're only going to be collecting safety and PK information for these patients, but 4 to 12 study patients are going to be assessed for efficacy as well as the safety. The dosing is going to be conducted based on weight, and then the patients who will complete the Neptune study are going to be eligible to enter the ELARA study, which will be ongoing. Primary outcome of the study is the CGI. This is the outcome that we received the approval from the FDA to use as a primary objective. We're also going to be measuring sleep. We've learned many lessons from the STAR study. We know that actigraphy is probably not the most tolerable device. We've done a lot of search what's going to be most tolerable, and I think we came up with a design or with a device that is going to be tolerated well by kids. And we're going to have other specific domains for the study that measures the other functions as well. So in order to qualify for the study, age is going to be 4 to 12, the CGI score of 3 and more. Molecular diagnosis of Angelman syndrome. We had a lot of kids in STAR study who came with a confirmed diagnosis. However, if the diagnosis needs to be reconfirmed, we will be providing these tests for the child. And as I mentioned before, the weight is going to play a critical role in determining the dose. What's exclusionary? The children who have uncontrolled seizures, unfortunately, are not going to be allowed to participate in the study. They will have to tolerate the sleep device for at least 7 days before the baseline. At risk, the kids who are at risk of harming themselves, and some other prohibited medications. Benzo specifically, although benzo is not allowed to be used continuously, we do allow some of the use benzo as sort of the rescue medication. We also allow CBD, knowing that Epidiolex is now confirmed and approved in US. The study is going to be monitored by the 2 committees. One is the Clinical Trials Steering Committee, and the other one is Independent Data Monitoring Committee. So separate group of people are going to be monitoring studies every 3 to 6 months on an ongoing basis, just to make sure that we have no safety concerns. So as promised, the site location. This is the sort of the ongoing process as we speak. The site feasibility is being conducted. We will have approximately 10 sites in US. Sites that are confirmed is Phoenix and Media, Pennsylvania. These are sites that we expect are going to be opening soon, very soon. Chapel Hill, North Carolina is also the site that we know is going to be open sometimes in probably September, October. The other sites we are pretty certain that we're going to have the sites open as well. So the goal is really to get the, if not the Angelman clinics, which is our preference. Obviously, we're looking for the physicians that have a lot of experience with the Angelman syndrome, but we also need to make sure that the sites are distributed evenly across the US. We know how many patients travel. We do provide the travel assistance, but the distribution of sites across the US is an important factor in selecting the sites. As I mentioned, Australia is going to be part of the study as well. We do have one site now identified in Australia. We're looking for possibly another site. We're going to have sites we think we're going to have sites in Germany and Netherlands and possibly in Italy. And we do have a site in Israel, Sheba Medical Center, who was also part of the STAR study. Site list is changing. We will be adding more sites. As I mentioned, we will have in total 15 sites globally, and most of the sites are going to be in US. I think this is my last slide. There are many PIs, there are many doctors in the audience yesterday and today who made STAR possible. We continue to rely on their guidance and their assistance. We're incredibly grateful to the patient population and PIs for supporting us in doing the study. Any way we can alleviate the burden of traveling to the site or making the study easy, we certainly here to listen. If you would like more information about the study, we have a booth set up outside, so you can get more information about the study. It's going to get published soon on this clinicaltrials.gov, and we will have a separate website for the Neptune study where you can get more information and get a pre-screened for the study. So with that, I'll take any questions. Okay, so if there are no questions, thanks so much.
Thank you.
Okay.
I'd like to welcome up to the stage from Ionis Dr. Frank Bennett, who's the Senior Vice President of Research. Clicker.
Okay.
Green to go.
Green, good, thank you.
Go.
So I'd like to thank the organizers for giving us an opportunity to speak today, and I'd like to thank you for being here. Over the next few minutes, what I'd like to do is just describe some of the work that we're doing to find a potential therapy for Angelman syndrome patients. And one word that you will have to learn how to say before you leave the building today is oligonucleotide. I think it's the first time you hear it, it's a hard word to say, but it's, I think as you'll hear in the next three talks, it's an important a potential therapeutic opportunity for Angelman patients. So just to give you a very brief introduction to Ionis, we're a company that's been around 30 years. Originally we were called ASF Pharmaceuticals, and we changed our name for obvious reasons a few years ago. We're located in Carlsbad, California, which is near San Diego. And we have about 500 employees, and we're a little bit unique in the biopharmaceutical business, in that 100% of the people in the company are focused on one technology, which is antisense oligonucleotides. And it's been the focus of the company for 30 years now. And so it is something that we live and breathe every day, and what our job is to find applications of the technology that could benefit patients going forward. Just to highlight is that antisense technology is really a new technology. I'll show you in a minute that it's a very efficient drug discovery platform, and it's we view ourselves as being a leader in innovating this technology and bringing it forward to patients. And it really is the poster child of precision medicine, in that we're going after the specific genetic defects that are causing a lot of the diseases, including Angelman's, as an example. So just to highlight what an antisense drug is most of you are aware that DNA is the material in the cell that codes all our genetic information. And then DNA is translated or transcribed into an intermediate molecule called RNA, which is then translated to a protein. And protein is the building blocks in the cell that sort of carry out our normal function. Most traditional drugs, things like statins or aspirin, or most traditional drugs, target proteins as their therapeutic target, and so they either bind to the proteins, such as small molecules, or newer generation technology called antibodies. Monoclonal antibodies is widely used in cancer and inflammatory diseases like rheumatoid arthritis. Both those types of technologies really do, the focus of them is on targeting proteins. What we have been working on is a technology that targets the RNA. And so by targeting the RNA, you, in general, can prevent the protein from ever being expressed. So we have a drug that binds to the RNA that's recognized by enzymes in the cell that chew up the RNA. And for most of our drugs, that results in a lack of production of the protein. As you'll hear in a second, for Angelman syndrome, our approach is to do the same thing. It binds to the RNA, chews up the RNA. But in this case, it's the antisense RNA, and I'll explain that in a second, that results in increased production of the UBE3A protein that you heard about a few minutes ago. So it's a little different approach, but it's really trying to get at the heart of what's causing the disease. And so it's not theoretical. There today are 7 antisense drugs that have been approved for use in patients, and they include, most of them are focused on rare diseases that include high lipids, other neurological diseases. In particular, one I'll highlight for a second is Spinraza, that's used to treat another neurological disease called spinal muscular atrophy. And the reason I highlighted that is that, similar to the way we're thinking about delivering a drug for Angelman patients, it's being delivered by an intrathecal injection. So it's what's called a lumbar puncture, and it's very similar, if anybody's ever gone through an epidural, very similar procedure to doing an epidural, where you're inserting a needle into the base of the back, below where the spinal cord is. So there's a space here. We're not going to hit the spinal cord. and you inject the drug. It takes 10 to 15 minutes to go through the procedure. It's been very well tolerated. We've used that for nusinersen or Spinraza is being delivered to patients that way. And today, over 8,500 patients in the world have received Spinraza. So it's not Star Warsy. It's a technology and a route of delivery that is being broadly used today for delivering medicines to patients. So just to highlight is that I've been working in the pharmaceutical industry now for 35 years, and so I've seen the industry evolve. But really, the steps in the process have not evolved. And so if you sort of break down the steps in more traditional drug development, the early stuff is the work that you've heard a lot about this morning from Stormy and sorry.
Mr. Philpott.
Ben, about some of the early basic research that's being done to help define what causes the disease and understand the biology of the target. The next step is then starting a drug discovery program based on these findings. So it's really a community. We have to work very closely with the basic scientists to be able to develop drugs. And it's one that neither team can do this alone. We have to work as a community. And that results in identifying a drug product that then has to go through a whole series of safety studies to make sure that the drug we're going to give to patients ultimately is going to be safe and well tolerated. And then it goes through a series of clinical studies. So the first clinical study is a phase 1 clinical trial that's primarily focused on safety of the drug. So the main goal of the phase 1 or the first trial in patients isn't to show efficacy. It's really to show that the drug is safe. And it's important to understand that. It's generally a short-term study, and the number of patients that are going to be exposed to the drug is going to be limited. The second study is the phase 2 study. That's when you begin to get some data on the efficacy of the drug, that it has a benefit. But again, the primary purpose of the phase 2 is to collect more safety data. And then the final stage is phase 3, where you're really trying to demonstrate efficacy. And the Ovid drug, as you heard about a moment ago, is at this stage in the development pipeline. So just to give you an idea of how we're approaching Angelman syndrome, you've already heard that Angelman syndrome is due to loss of a gene called UBE3A. And it's a paternally silenced gene, so the father's copy of the UBE3A is sort of silenced or locked up where it can't express the protein. And really the protein production is depending on the mother's copy of the gene. If there's any kind of genetic change, such as a deletion and a variety of other mechanisms that can silence the mother's copy of the gene, there's no UBE3A being produced in neurons. And so our approach is to use antisense oligonucleotides to unsilence the father's copy of the gene. And the way to think about this is that there's what's called an antisense transcript. That's the UBE3A ATS antisense transcript. That is, think about a train moving down the tracks this direction, whereas the normal UBE3A train is moving this direction, and there's a collision. These two polymerases, the enzymes that transcribe the DNA into RNA, are colliding on this track, and that blocks the production of the UBE3A RNA. And so what we're doing with an antisense oligonucleotide is to stop this train from going down the track to allow this train to continue to be expressed. And so it's a way of sort of what do you call it on the train track, where you're shifting the rails of the train so that they no longer collide. And that's the approach that we're taking with the antisense technologies. We're causing this part of the train to stop here so that this can complete and make full-length UBE3A protein. And so I think many of you are aware that we've been collaborating with Art Vedet's lab for a number of years to develop this concept. And that resulted in a publication a few years ago highlighting mechanistically that antisense is a technology that might be useful for treating Angelman syndrome patients. And just to highlight a couple of points of it that were published in the paper is that with an antisense drug, we were showing that we can decrease the production of this antisense transcript shown here and increase the production of the UBE3A protein. So it really does support mechanistically that the drug is doing what it should be doing. That was in cell culture. We've also shown the same thing in mice that are engineered to express a reporter gene. But what we show in the mice is that you can decrease the production of the antisense transcript in different regions of the brain, cortex, hippocampus, or spinal cord. And then at the same time, see an increase in UBE3A protein. So it really was a proof of concept study that we did. Very important. Without that, we wouldn't have been able to go forward, but it was really a proof of concept. We didn't discover a drug out of this process. So the next step is discovering a drug, and I think many of you are asking, that was published 4 years ago. Why is it taking so long to develop a drug? And just to say that it's a complex process to identify the drug. And what we've done is design thousands of oligonucleotides, so thousands of these little pieces of RNA to bind to the antisense transcript and screen those in cell culture experiments to show which ones have the desired effects and which ones have the no activity or undesired effects. Those then go into animal models to show that they're safe, and then also that they do engage the target and produce the effect that we like. And ultimately, you end up with a single drug that goes into safety studies, IND-enabling studies that the regulatory agencies require you to do. And so it's been a long process, but I think we're seeing the light at the end of the tunnel from this process. So just to highlight some of the results that have come out of the screening exercise, this is one lead compound that was very effective at reducing the antisense transcript. So this is normal level. This is after treating in cell culture with the antisense transcript. And you see that with looking at the effects of the oligonucleotide shown here in green, we're getting very close to what the normal level of UBE3A expression is, which is in the black here. So we found a large number of oligonucleotides that work very well in doing this. The challenge has been how do we translate this going forward into patients. These are cells that are cultured in a petri dish, and we really wanted to show that they have some benefit in a living organism. The human and mouse sequence are not well conserved, so the oligonucleotide or the drug that we identified to target the human sequence does not work in mice. And so what we had to do, and that's what's taken so long, is engineer mice to express the human gene. And in doing so, we've been able to document that the drugs that we've screened are producing the effect that we desire. That's taken a couple years, and it's I think, added a lot of complexity to the program, but we're there. We've accomplished this. And so the next steps, and these are being done in parallel, is continuing to investigate mechanistically how the drug is working. That's not gating. I think we have enough information today to go forward. But really the next step is to demonstrate safety that would allow clinical trials to start. So that's doing toxicology studies in different species. At the same time, we'll begin engaging with the FDA and other regulatory agencies, such as in Europe, to design the clinical trial and discuss with them what are the endpoints that we should use, what are the doses of the drug that we should use, et cetera. And then the time is we continue to collaborate with the physicians, the patients in these Angelman clinics, to help us design the best clinical program to ensure the best chance of being successful at the end of the day. So I'll just very quickly, because the next two talks will really highlight this, is that there are some gaps in our knowledge. And to be able to execute on our plan, it's really important that we get more natural history data. And so the next two talks will really focus on that, is that there are a number of natural history studies that are ongoing or about ready to start. And your participation in those natural history studies will ensure ultimately that all companies, not just Ionis, but all companies will be successful. Because one of the things that we've worked out is developing a consortium where this is what we call pre-competitive studies, where we're all helping to do the studies, and the data will be shared among all the companies that are participating, as well as with the Angelman community. And so with that, I'll just conclude that we've made great progress in identifying a potential drug candidate. The transgenic mouse slowed down the project, but we've now solved it and are moving forward. And ultimately, as you'll hear about, your participation in these natural history studies will be instrumental in having the success and ultimately bring a therapeutic product forward. And so with that, I'd just like to thank my colleagues at Ionis and Biogen academic collaborators who we wouldn't be here today talking about this program without their help and your help in funding the work in their labs. And then also the Angelman foundations that are supporting you guys and also supporting the companies that the work that we're doing. And thank you.
Thank you so much. Perhaps what would be best is if we left the questions for the end of the session. So Dr. Bennett mentioned there will be a quiz on how you can pronounce oligosense antinucleotide at the reception tonight, and you'll be given bonus points for how many drinks of Kentucky bourbon you've had prior to. I'm kidding. All right, I'm welcoming to the stage right now Ina and Michelle from Roche, who are going to give us an update on where Roche is in their Angelman project.
So thank you very much for that introduction. I want to start by saying, well, who I am. I want to just introduce myself. My name is Ina Brünig-Träbert. I work in Basel at the Roche headquarters in Switzerland, and I took over the leadership for the Angelman Development Project at the beginning of this year. So I really want to start by saying how excited I am to be here at this meeting. It's really an unbelievable experience for me personally. It's a big milestone in my steep learning curve about Angelman as a disease and what it really means for families to get that diagnosis and to live with it every day. So as a caregiver of a child with a diagnosis of autism, it helps me relate to the things that you go through on a day-by-day basis, but I appreciate the differences. And it's really a major milestone for us because everything we do at Roche with regard to this development program is centered around a deep understanding of the disease and the patient perspective. So it's really important for us, we care about this, and this is why this meeting is really very important for me and for us at Roche. So thank you, we're very grateful to be here. I just want to start by basically introducing the company and the project itself, and then I will hand over to Michelle, my colleague who is the clinical lead for the Angelman Development Project, and she will tell you about the news that we have with regard to our efforts to bring a drug to the clinic really as soon as possible. This is our disclaimer, so everything we say here today is our own opinion and not that of Roche. So just generally speaking, Roche really has quite a rigorous approach to drug development in rare diseases in general. So it's quite different actually from how we develop drugs in non-rare diseases, and this is a very, very targeted approach. So Angelman syndrome is quite an impressive example of how we build our expertise and knowledge around the biology of the disease, and that builds the basis for then designing molecules that have a potential to be disease transformative. So it's really a nice example of that. While we are trying to identify the right molecule and further optimize it and characterize it, we have quite a lot of efforts ongoing in achieving a very deep understanding of the disease, which will help us to understand what's meaningful for patients and families. Because this has to be the center of how we will ultimately design our clinical trials. Of course, clinical trials have to be approvable. They need to be enabling to bring the drug to market ultimately, but they also have to be meaningful in terms of what they measure, in terms of what families really need. So we have developed a piece of work that we call a disease concept model, that has been done in collaboration with many of you, probably with patients, caregivers, with clinicians and experts in the field, which helps us to find out what a treatment actually would need to do to families, what would it need to do to the patient in order to be meaningful. So Michelle will present some of this data in a few minutes. And the other really important aspect to us is collaboration. So obviously this can only be done through really strong collaborations between industry and the community, but also amongst industry. So we really feel quite strongly that we absolutely should join forces here, and we are doing that already with some of our colleagues and some of the other industries to work together because the goal ultimately has to be to get data to then inform clinical trial design and bring drugs to patients, to you, as fast as possible. So we really want to try and keep competitive considerations aside as much as possible to advance the field faster. So this is a slide very similar to the one that Frank just presented, and it's really just highlighting the type of molecule that we are working on. So as many of you know by now, the genes obviously are encoded on the DNA, and in a cell-specific manner, pieces of RNA are made from DNA. So those are copies of genes. And since scientists have found ways to interact with these RNAs in different ways, a number of new treatment options have come up. And one of them is the ASOs that Frank talked about and others before. So at Roche, we have a few examples of those already in our portfolio. So for example, for the treatment of Huntington's disease, which is another rare disease, we have such a molecule that interacts with the DNA, with the RNA to downregulate a certain protein that is actually causing a disease. And in the case of spinal muscular atrophy, which is again another rare disease, we also have a molecule that interacts with such an RNA, but in a slightly different way. So in the case of Angelman syndrome, obviously, what we're trying to do is to interact with this piece that is called antisense RNA that most of you have heard about now a couple of times, in order to bring back the UB3A protein expression in neurons, which is what's missing in Angelman syndrome. The molecule, so this is the slide that's showing actually how the molecule works. So, and I just repeat it, even if it's repetitive for many of you. So obviously in Angelman syndrome, the mother's copy of the gene that's encoding UBE3A is lost or defect, and the paternal version of that gene, so the one from the father, is silenced by the piece called antisense RNA. So our molecule interacts with that piece, that antisense RNA, to unblock, unsilence the father's copy of the gene. So where are we with regard to our drug development project, which is obviously an important question for many of you. So we left behind most of the discovery work on our molecules, and since this is something that's still going on even into the clinical phase, we will further characterize and work on our molecules, but the phase that we are currently in is really animal testing phase. So we are in the preclinical phase, and we are testing the molecule in animals for its safety and tolerability. So a piece of good news that I can share is that one of our lead molecules have been tested in a study in cynomolgus monkeys, and we were able, with a single dose of this molecule administration, to bring back the UB3A protein for a prolonged of time, for a duration of 85 days after a single dose, which was quite a nice thing to see, of course. We're really happy to share that piece of news. Obviously, now a couple of other things need to happen and be in place before we can then ultimately go to the clinical development phase. But one thing that we do in parallel to prepare for the clinical phase is what we call endpoint development. Now, endpoints are measurements that we do in clinical studies to show that the drug is actually doing something beneficial for the patient, and that will help to measure whatever happens as response to drug treatment. And it's also something that we have to agree with the regulators on, that this is acceptable for approval. So it's very important that we get this piece right, because if we don't have it, we won't have successful clinical trials. So I will pass on to Michelle now to talk to you about our work in endpoint development.
Hello everyone, I'm Michelle, and I'm a pediatrician and a clinician at Roche, and my responsibility is to really think about the parts of designing a clinical study that involve you, that involve families. So that's really what we're focusing on at Roche now, and that really, a lot of that happens actually before any drug is involved. So what Ina has been talking about is what happens before that line on the slide. So a lot of that is what you've heard earlier today. And what we're really focusing on now is what's going to happen after that line. So what is happening in the future, and what are our forward-looking thoughts. And so this is something that we are working on very actively now. And so there are a number of steps that are important on the journey towards clinical trials. And as I say, many of them happen and are actually independent of drug, and they are very important for a number of companies and for also academics and clinicians who are working with you every day. And so that's something that we're working on now, and that involves most importantly, it involves you and involves your children. It involves the academics, the investigators that you've heard presenting earlier, the regulatory authorities. So the FDA is a very important part of this conversation, and we will be engaging more with them as we go forward. And in terms of the clinicians that you heard from the Clinic Network, as well as other companies, there's a number of questions that we need to answer on our way. And they start with, do we really understand Angelman? How do we know how to measure aspects of Angelman that are important to the families? And how do we use that knowledge to design clinical trials that actually are what you need, and that can answer the question of whether a particular drug is actually helpful to your families. And then we need to importantly understand the safety of any new drug. This is a crucial point. And how do we do this in a collaborative way? That is what is the best approach for families. So one of the steps along this way that was started very early, actually 2015 or so, was actually working towards this disease concept model. So this is a piece of research that is directly linked to families, and it's about understanding what aspects of Angelman should an effective therapy help. What would families like to see changed by a therapy? And so this involves a number of steps, and the most important step is actually conversations and interviews with families. And so that was a piece of research that was done in collaboration with Roche and the ABOM, so the Angelman Biomarker and Outcome Measures Consortium that has been involving ASF and FAST and a number of other companies. And we really did that to understand what to measure and how to measure this in future clinical trials. That is a very detailed piece of research where there's a report that is actually available, and it's very extensive. And we're now summarizing that into a report, into a manuscript, into a paper that we're going to publish, and that will be public information as well. And so what we do is, with the output of that, we found that there were a number of very important aspects that families highlighted as being things that would be critical and important for them to see a therapy change. None of these will be surprising to you, they're part of your daily lives, but it is important for this to be really described well so that the FDA and other people, researchers and clinicians are also aware and can refer to this. And so these are aspects that, now that we know that these are the key things that we can take forward, we started to take these and to understand how we could measure them in the clinical trials. And so what we did was to look at the assessments that are done in clinic, and you'll be familiar with these from your visits. So the Vineland and the Bailey scales, which many of your children would have done, and also a behavior checklist that is widely used. And we looked at whether there were any gaps in what these scales capture versus what is important to families. And what we see is where you see the orange color is where there are gaps. So sleep is something that families have said is very important for day-to-day life and for quality of life. And also seizures was something that also was mentioned to be very important to families to modify. And so these existing scales don't address those questions. And so we saw that there's a need to actually learn about that and to find ways to study that in a clinical trial so that if a therapy comes, it actually addresses that. So we actually are collaborating together. So Roche and Genentech are the same company. So in the US you will hear Genentech mentioned, but it's one company, so that's us. And we're working together with Biogen and Ionis. So Roche is the study sponsor. That means we're responsible for running the study day to day. But this study is co-funded by 3 companies, and we will be sharing the data among the companies. The benefit of this to you is that instead of us all running separate studies and asking you to take part in lots of separate things, we're actually trying to just run one study that we can all take the data from, and so that is actually the best thing from your perspective, as well as the fact that we are sharing the data that is overlapping with the natural history study that many of you might be aware of, and also with an MRI study that's being run at UNC at Chapel Hill. So a lot of these data will actually be available publicly, and they will be shared between the companies. And so this is a 1-year study, and it involves mostly activities in the home. There are a couple of visits to clinic at the beginning and at the end, but most of the activities that happen are in the home. It involves about 40 patients with Angelman as children, so aged 2 to 12, and then there will be 10 adults.